various compartments inside mitochondria. Those are largely new or improved inventions. They also have to acquire lipids from the ER to build themselves, although they have some of their own lipid biosynthetic machineries, nuclear encoded. But here, we've also been able to acquire for our mitochondria dynamic behaviors that allowed them to evolve in a way that was integrated and tied in to other pathways in the eukaryotic cell. So these are important new inventions. So what I'd like to start with is to tell you a story that spanned my lab probably 15 years on dynamics, mitochondrial dynamics, from the discovery of these components to some of the mechanisms that we've uncovered and how they work to build the machines that divide and fuse mitochondria. So here again is a yeast cell, a wild-type yeast cell, and you see again mitochondria are quite connected. In fact, the relative rates of mitochondrial division and fusion we discovered are roughly balanced. So at any time, if you take a snapshot, it's a really low copy number structure. And this is not a weird oddity of yeast. You will see this in human cells too, and most of the human cells in your body, although there's some exceptions, where the mitochondria are organized into these long tubular connected reticular structures. Now, knowing that the rates of fission and fusion were roughly equivalent gave rise to an idea in my lab about how mitochondria might look if you could selectively mutate genes that blocked fission or fusion. And in fact, the changes in the morphology of mitochondria, when you do that, which we predicted, turn out to be true. So if you block mitochondrial fusion, and in yeast there's exactly 3 proteins that are essential for this process, so the machines aren't complicated by nature, division goes on. So while somehow they're linked, the levels of their rates are linked in ways we still don't understand, you can disconnect them. And you take this connected structure, division goes on, and you disconnect it. So this is what maybe people still think of as mitochondria. I don't know. On the other side of the equation, if you block division, again, there's 3 proteins that are essential, simple. Fusion goes on, and this connected structure you see here connects up even more. So clearly, the balanced rates and continuation of co-processes of division and fusion are really essential for maintaining this kind of steady state structure. And what yeast also told us was the real fundamental roles these processes play in cells. So if you block mitochondrial fusion or division, you start really screwing up with the transmission of its chromosome. In the case of fusion, you get complete loss of mitochondrial DNA. So fusion is absolutely essential for the maintenance of mitochondrial DNA and its transmission. So, and certainly division is really important for that. And here I want to point out that especially in the case of blocking division, you see a dramatic defect in the distribution of mitochondria. So here again in the wild type cell, at the cortex where they're positioned, they're nicely spaced out, right? So if you know, at any given point around the cell, there's some mitochondria nearby if you pointed at it. Here, if I focused on the top of the cell, it would be bare. And especially in this cell type, where you divide by growing a bud and transmit in a really directed way, this is important to spread it out for efficient segregation into daughters. And in fact, these phenotypes that you see here are recapitulated in human cells when you make mutations in the conserved components of division and fusion that we identified in yeast. And in fact, mitochondrial division becomes especially important in highly polarized human cells like neurons for the maintenance of neuronal cell physiology and health. So these 2 events maintain this really nice but spread out connected structure and are required for the essential, the very heart of mitochondrial function, its respiratory function. They have to be together, okay? So you have to have them together. In fact, we think they co-evolve together. So one of the things that's pretty remarkable is that the core components of division and fusion, as I'll talk about in a minute, are highly conserved all the way through humans. The human processes, the human components have been linked to they're much more highly regulated And in fact, dynamics in human cells, not so much in yeast, have been linked to quality control, for example. And I just want to demonstrate this in a very simple way, how it works. If this is a mitochondrial tubule, and these red dots are wild-type mitochondrial chromosomes, and the blue is a mutant, you can see division could give rise really to 3 different types of products. One that lacks genomes, one that segregates away from the wild type, which is a buffered situation. Okay, so fusion buffers problems. You could segregate a defective genome away, which could ultimately result in mitochondrial dysfunction and loss of membrane potential. And work from many labs, including, for example, Richard Yule's lab, has shown that when this happens and you get a decrease in membrane potential, that triggers a process called mitophagy, which is the active, selective degradation of mitochondria wholesale using autophagy. Selective autophagy is the mechanism. Work from my lab reconstituting mitochondrial fusion in vitro shown that membrane potential is an intrinsic component of the fusion reaction. So once you get a situation like this, these mitochondria can no longer join back to the herd. They're forever cold and degraded. So it's a really clever way, again, harness dynamics, this continuous process of dynamics for a quality control pathway. When stress happens inside the cell, and by stress I mean different kinds of pathology, it's been fairly well documented now. I have a table like you do, Vamsi, for a uniporter, where this causes a massive increase in division by mechanisms, some which we know, some which we don't. A shift to this fragmented state. So not only does fragmentation promote mitophagy, but on an extreme stressful situation, it goes wholesale cell death. Permeabilization of the outer membrane, release of cytochrome C, cell death is linked to an increased level of mitochondrial division. Again, another stress pathway. And in fact, mutations in 2 of the conserved components in humans that are required for mitochondrial fusion cause 2 distinct types of neurodegenerative diseases. So underscoring the real importance of dynamics in human health. And I also like to just point out here what's increasingly becoming clear in human cells is the division and fusion rates are very much linked to metabolism, the metabolic state of the cell. So there's fine tuning, of these components. Really cool, cool stuff. So amongst these 3 conserved components, they all belong to a single protein family. That was pretty cool when we first discovered it. We're like, wow. And the family are these dynamin-related GTPases. So they're GTPases. They have canonical GTPase domains. But in addition, they have other regions that facilitate both intra and intermolecular actions. So these proteins build structures. And they build structures specifically in association with membranes. This is a prototypic member of the family, dynamin-1, a collaborator of mine, Jenny Hinshaw at the NIH. This micrograph shows that dynamin-1 can build these really beautiful helical structures. Dynamin-1 is involved in endocytosis, in the scission of clathrin-coated pits from the plasma membrane, a fission reaction of a different sort. But when these structures form in association with membranes, they change the shape of membranes. And it is thought that they act in that capacity as mechanochemical enzymes to not only change membrane shape, but in a dynamic way to produce force that, with additional conformational changes, that causes, for example, membrane scission and membrane fusion. So built into these structures, assembling is a stimulated GTP hydrolysis, so they serve as their own gaps. And in fact, that's part of the power stroke. So I want to tell you briefly, again, we took advantage of the fact that there were 3 components, and over the years, purified these proteins and reconstituted a lot of the reactions that occur during mitochondrial division in a test tube, culminating in a crystal structure of the dynamin itself, or a very good model for it, actually the endocytic dynamin. Again, the machine that builds mitochondrial division. So before I show you the kind of details of the biochemistry and so on, I want to show you what this division machine looks like in vivo. It's in green here. This is a functional GFP fusion. In red are mitochondria in a yeast cell. This is a yeast cell here. And you can see at steady state, in this cell type, it's already assembled. And I want you to really just look at that structure right there. That's where a fission event takes place. And in fact, every single division event that takes place in vivo is linked to one of those structures. That was pretty compelling to us, that they were the heart of the division machine. And so over the years, we've discovered how assembly is regulated, how it finds its way, this machine, to the mitochondrial membrane instead of some other organelle, how it does